Hi, thanks for joining me on our discussion over the process of homeostasis. The assigned chapters are 30 and 31. So for chapter 30, homeostasis part 1, we're going to talk about osmoregulation and temperature control. Homeostasis is a concept used almost exclusively in physiology, which would be Biology 2424 here at Rose State College. And it refers to the body's ability to adjust internal functions due to the environmental changes. Homeostasis is a term coined by the 19th century Harvard professor by the name of Walter Bradford. We can use body temperature as an example for how the constant internal environment tends to revolve around a given set point, like human body temperature. You probably recognize the normal body temperature of humans as 98.6 degrees Fahrenheit, which is converted to 37 degrees Celsius. Any deviation from this physiological set point will activate the negative feedback regulation process in order to return the system to this normal temperature. Negative feedback control are systems that work in coordination to return the regulated variable, which is temperature, back to its normal set point. There are sensors that detect the changes from the physiological set point in the internal environment. There is a central nervous system, which would be the brain and spinal cord, if you're talking about vertebrates, that would receive this information and then determine the response. And then effector organs. Effectors produce the response that restores the variable back to normal levels. This is usually achieved by muscles and glands. Homeostatically controlled systems make the adjustments required to lessen the impact of major environmental disturbances. Many factors within an organ's internal environment are regulated by homeostasis, like blood pressure, the amount of blood flow to an area, your breathing rate, the pH of your blood, urine concentration, and the release of hormones. Water and the associated osmotic concentration based on the minerals or electrolytes in that water also have to be tightly regulated in order to prevent death of the organism. The volume of an animal cell can increase or decrease depending on its fluid environment. A solution is called hypertonic if it has high salt content, or hypotonic where there's low or no salt concentration. These types of extreme environments can have a distressing effect on the delicate cells and their metabolism. Animal cells have a selectively permeable cell membrane that regulates ion concentration by managing salt and water movement across the membrane. Multicellular animals can control ion and water balance inside their cells by using protein pumps and ion channels embedded in the plasma membrane, which add function to their barrier between the ICF compartment and the ECF compartment. So extracellular fluid and intracellular fluid are what ICF and ECF stand for. The ECF compartment consists of plasma and interstitial fluid, which contains amino acids, fatty acids, sugars, salts, enzymes, hormones, neurotransmitters, and even waste products that are released from the cells. In this example, we can see how water can move passively across the plasma membrane by osmosis. This is defined as the diffusion of or movement of water only. Animal cells placed in hypotonic solutions will expand and swell as they take on water. If this movement continues unchecked, the cell will burst. This is a process called lysis. The concentration of water is expressed in moles per liter in this example. The term tonicity is used as a suffix describing the solution's effect on the cell itself. Isotonic solutions create balance with respect to the cell. This is where both ECF and ICF compartments have the same osmotic pressure. A hypotonic solution has higher osmotic pressure outside the cell and are osmotically active. And hypotonic solutions have lower osmotic pressure than the cell. Therefore, water rushes into the ICF compartment, causing the cell to swell. Marine invertebrates live in a high salt oceanic environment and have evolved methods to maintain salt and water balance. Most are in osmotic equilibrium with their seawater environment because their body surface integument is permeable to water and salts, which means their internal body fluid concentration will change along with any changes in their environment. Luckily, for billions of years, the Earth's oceans have been relatively stable. When an animal has limited ability to withstand changes in salinity, they are characterized as stenohyles. Conditions along the coast and estuaries are much less consistent, so animals have to cope with abrupt changes in salinity and have evolved to tolerate these variations and are characterized as being euryhalines. Salmon are an example of a euryhaline. We use the term osmoregulation for the animal's ability to resist dilution of its own body fluids due to living in dilute seawater. An animal that is hyperosmotic to its environment has to regulate its internal water balance. Kidneys or antennal glands found in aquatic crustaceans can help the animal maintain a higher concentration of salts by excreting excess water. Salt-secreting cells in the gills of fish actively remove ions from dilute seawater in order to counteract the loss of salts flowing across the gills into dilute water environments. Only 3 animal phyla can make urine hyperosmotic to their blood plasma. That includes insects, birds, and mammals. Freshwater fish have developed many adaptive mechanisms to prevent salt loss and unload excess water from their bodies. In panel A here, water that enters the ECF compartment by osmosis across the gills gets pumped out by the kidney in the form of very dilute urine, which is released in large quantities. Salt-absorbing cells found in the gills help move salts from the water into the animal's blood plasma. Freshwater fish use little ATP energy on osmoregulation due to the efficiency of their kidneys. Clams, crayfish, and aquatic insect larvae are also hyperosmotic regulators with similar mechanisms to freshwater animals. Amphibians that live in freshwater use their skin to transport sodium ions and chloride ions into their interstitial fluid compartments. Urea helps maintain a hyperosmotic internal environment and minimizes water loss across their highly permeable skin. Salt and water balance in marine fish. In this example, marine bony fishes maintain salt levels in their body fluids at about 1/3 of that of the seawater that they are living in. This is called hypoosmotic regulators. Water concentration inside a fish is higher than in the ocean itself because the ocean is so salty. As a result, most saltwater fish constantly lose water across their gills and skin. Marine fish constantly drink seawater to compensate for water loss. And to get rid of excess sea salt, the fish's kidney excretes massive amounts of salt into its urine. Special salt-secreting cells in the gills pump the salt back into the sea. Marine fishes consume only enough seawater to replace water loss. Elasmobranchs, like sharks and rays, achieve the same osmotic balance with their environment by a different mechanism using urea and trimethylamine oxide, or TMAO. Shark kidneys conserve urea, which is normally a toxic waste product, and accumulates this urea in their blood and body tissues until there's no more osmotic difference between them and the seawater. This establishes a water balance between the animal and its salty environment. Marine reptiles and birds have salt glands next to their eyes, which help them secrete large amounts of salt, which gets ingested from their diet. These salt glands excrete highly concentrated sodium chloride, which is table salt, that can be up to twice as concentrated as their saltwater environment. The production of salty tears is very important to these animals' success, since their kidneys can only produce very dilute urine and cannot manage all the salt that they take in from their environment. In this example, the unicellular freshwater paramecium shown here displays a special excretory organelle called a contractile vacuole. The lines are pointing to these structures. These are small spherical structures that are not considered true excretory organs. Ammonia and other nitrogenous wastes can diffuse across the cell membrane directly into their watery environment. Contractile vacuoles act as an osmotic organ, creating a water balance by expelling the excess water that enters the animal due to osmosis. Contractile vacuoles are absent in marine animals because they are isoosmotically balanced with their saltwater environment. Here is Flatty Platty, the platyhelminthes from one of our lab experiments, and is an example of the invertebrate filtering structure called the nephridia. This is common in phylum Annelida. These are simple tubular structures that produce filtrate. These fluid secretions come from the blood, which then exits the animal's body in the form of urine. This is the most common design used to maintain osmotic balance, and it uses a flame cell system or protonephridia as a simple arrangement for observed in the acoelomates and in certain pseudocoelomates, like our example here. This protonephridia system, shown in green, there's a pointer and it's labeled protonephridia, so you can see my cursor right here. This is made of highly branched ducts that extend out into all parts of the body. Fluid enters the system through specialized flame cells where rhythmical beating of a flagellar tuft of cells creates a negative pressure, and that draws the fluid into these tubules. As fluid moves through these tubes, water and metabolites are recovered by reabsorption, and wastes are expelled through excretory pores. Nitrogenous wastes like ammonia diffuse out across the animal's integument. Flatworms have no true circulatory system, so the flame cell system is extensively branched throughout the animal, which provides excess surface area for better efficiency. Protonephridia creates a closed system. Metanephridia is a more advanced form, which is an open system found in mollusks and annelids. The tube is open at both ends and allows fluid to be swept into the tubule through a ciliated funnel-like opening called the nephrostome. This is surrounded by a network of blood vessels that reclaim water and valuable salts. The basic process of urine formation in the tubule remains the same for protonephridia and metanephridia. This process ensures the removal of waste without the loss of useful molecules. In the arthropod kidney, there are two basic types of arthropod kidneys which are uniquely used for excretion. First, we see the paired antennal glands of crustaceans. These are located on the ventral portion of the head and is one form of the basic nephridia. They lack open nephrostomes, which are the funnel-shaped part of the metanephridium that works to push out wastes. In this example, the hydrostatic pressure from the blood forms a protein-free filtrate in the end sac. In the tubular portion, highlighted in green, certain salts are selectively reabsorbed or actively secreted into the filtrate. This system is similar to the vertebrate system of excretion as far as the sequence of urine formation goes. And then if we consider myriapods and insects, and some arachnids, such as spiders and mites, they possess another type of excretory organ called Malpighian tubules, which they open directly into the intestinal tract. These thin, elastic tubules form a closed system, and they lack arterial blood supply. Urine is produced by tubular secretion mechanisms initiated by active transport, which requires ATP energy, and they move hydrogen ions into the hollow portion of the lumen. Protein transporters move hydrogen ions back into the cells by an exchange process with sodium or potassium ions for chloride. In the earliest vertebrate kidney, it's tracked back to the formation of a true coelom, composed of segmentally arranged tubules, similar to the invertebrate nephridium just described. Each tubule opens into the coelom at the nephrostome, and the other end leads into a common archnephritic duct. This ancient kidney is called an archnephros, which here in the upper left-hand corner is the diagram for archnephros. This is similar to the segmented kidney found in the embryonic hagfishes and caecilians from phylum Chordata. From the earliest developmental history, it appears that the reproductive system developed alongside the excretory system and utilized the nephric ducts to move reproductive substances. In all of these examples, you can see reproductive organs alongside the excretion system. If we compare now the vertebrate kidney and its functions. So for the removal of metabolic waste from the bloodstream, these vertebrate kidneys also have major osmoregulatory function. The vertebrate kidney is part of many interconnected structures that maintain cellular homeostasis of salt and water balance. This is the primary organ that regulates volume and composition of all internal fluids. Each vertebrate group has a slightly different kidney organization, but all are going to have the same basic functional unit called the nephron. Urine is always formed by filtration, reabsorption, and secretion of internal fluids. These are directional terms that we'll look at in just a minute. The nephron does the work of filtering the blood, which is under arterial pressure from the beating of the heart. There are millions of these microscopic structures working to rid the blood of toxic buildup like urea, and they regulate salt-water balance as well as maintain proper pH. There is a round corpuscle region and a series of tubes which can be seen pretty easily in this diagram. Each segment of the nephron has a specific job to do in the process of forming urine from the fluid plasma portion of the blood. The renal corpuscle consists of a glomerulus, which is shown here in red. It's a capillary network that forms the surface area for filtration to occur. This delicate glomerulus is surrounded by a C-shaped Bowman's capsule that receives newly made filtrate. The renal tubules are divided into three parts. The proximal tubule, which is closest to the Bowman's cup, and it's completely permeable to water. This is where we can reclaim the majority of the water that gets filtered out of the blood. The proximal convoluted tubule turns downward into the descending straight portion that connects to the loop of Henle. This is where salts, mostly sodium, potassium, and chloride, can get removed from the filtrate. Finally, the distal convoluted tubule not only connects the nephron to the collecting duct, but is also the target of hormonal regulation that works to fine-tune osmotic homeostasis. Filtrate follows a path through the nephron where it passes into the proximal convoluted tubule, then to a thin-walled loop of Henle that extends deep into the kidney's medulla. This was highlighted by a darker brown in the previous slide. And lastly, through the final portion called the distal convoluted tubule. Filtrate then empties into the collecting duct that drains into the renal pelvis where urine collects and then moves through the ureters towards the bladder. Urine leaves the collecting duct in a very different form from the initial filtrate produced in the renal corpuscle because throughout the passage, some solutes like glucose and sodium are reabsorbed and others like hydrogen and urea are concentrated. Urine formation begins with glomerular filtration. The glomerulus is a specialized mechanical filter that uses blood pressure across capillary walls to produce a protein-free filtrate that collects dropwise into the C-shaped Bowman's cup. Small solutes can pass through the capillary walls, but red blood cells and plasma proteins are way too big to go through these tiny pores. After filtration occurs across the glomerulus, the filtrate begins its journey through the tubules and reabsorption occurs next. This is where we reclaim the majority of the water from the filtrate. Further along the tubules journey is where secretion can occur. And in this step, you can see that the arrow indicates movement of solutes back into the tubules for elimination from the bloodstream. Finally, the urine products get excreted out from the body. Water excretion by the kidneys works to regulate the osmotic pressure of blood. This means that when fluid intake is high, the kidney excretes dilute urine and saves salts. When fluid intake is low, the kidney conserves water and forms more concentrated urine with low volume. A dehydrated person can concentrate urine approximately 4 times their blood osmotic concentration, which is an important adaptation for terrestrial animals and prevents water loss while still being able to excrete waste products. Now let's move on to temperature regulation. Some animals can regulate their own internal body temperature, and they belong to a group of homeothermic organisms. Alternately, you may have seen reptiles that come out in the morning and sun themselves on a rock in order to warm up. These animals are called poikilothermic. Biochemical activities are sensitive to the internal environment, and temperature has a major impact on protein enzyme function. All proteins have an optimal temperature range and can become damaged or denatured when they are exposed to temperature extremes. Endotherms are just grouped in a general way. Any animal that is, ectotherms. I said endotherms. Ectotherms. Ectotherm is a general term used for any animal that is dependent on external sources of body heat. These animals have adopted behavioral adjustments that help them regulate their body temperature. Endothermy is used to describe any animal that can regulate their own internal biochemical processes that continue to contribute to basal metabolic rates and are able to increase their own body temperature outside of the act of shivering. Endotherms have an advantage and are not limited by the climate or temperatures in their environment, so they can populate habitats that would kill a poikilotherm. Animals that are small and have large surface area to volume ratio that results in high heat loss and explains why small creatures tend to decrease their activity in cooler weather. This graph shows how a small mouse maintains a constant internal body temperature even when its environmental temperature changes. Desert lizards exploit hour-to-hour changes in solar radiation to keep their body temperatures relatively constant. For example, in this diagram, in the early morning, the key is their exposure of just their head, which is absorbing some of the sun. And by mid-morning, they emerge from their burrow completely and bask in the sun with their bodies sort of flattened out. And as the day gets warmer, they turn to face the sun and reduce their exposure, raise their body up from the ground. In the hottest part of the day, they retreat to a burrow or they'll seek shade. If we use mammals as our example for endothermic temperature regulation and note that they have body temperatures consistently between 36 degrees Celsius and 38 degrees Celsius, which is maintained by a delicate balance between heat production due to metabolic activity and heat loss. Most of an endotherm's daily caloric intake goes into generating body heat. So in cold weather, endotherms need to eat more calories than ectotherms of the same size. Heat loss is a process of radiant energy, conduction, and convection caused by air movement across the skin or integument, as well as by evaporation of water. If an animal becomes cold, it generates heat by using its muscles and can decrease heat loss by increasing the insulating fat layers. If an animal gets too warm, it decreases heat production. You may notice many different animals in the wild napping during the heat of the day and increase their heat loss by finding shade or a pool of water. Certain mammals, like this desert kangaroo rat, have several adaptations for their hot, dry climate. Again, we see behavioral modifications like living a fossorial lifestyle by burrowing into the ground, or being nocturnal, where they're more active at night when it's cooler. Most notably about desert life is that there's very little or no water supply. These animals can derive all the required water from their food and by producing highly concentrated urine in a very low volume. Adaptations for cold environments. Mammals and birds use decreased conductance and increased heat production to survive the cold and maintain homeothermy. They possess thick fur and an undercoat which insulates and long guard hairs that keeps the elements off their skin. All birds have down feathers that function to conserve heat. On cold mornings, you may see birds sitting on a branch with their feathers all puffed out. They're creating a warm airspace between their skin and the thermal down feathers, which are not used for flight. Heat loss through the extremities, like the legs, a tail, ears, and nose of Arctic birds and mammals, are a source of rapid cooling. Some animals use warm arterial blood flow to warm up cold blood from the extremities in a process called countercurrent heat exchange. The countercurrent heat exchange prevents a well-insulated body from losing substantial heat through blood flowing along exposed limbs. And how this works is the arterial blood in the leg of an Arctic mammal or bird passes in close contact with a network of small veins, shown in blue, that have cold blood returning from the limbs. In this diagram, heat exchange occurs along the opposite flowing vessels, which transfers nearly all of the body heat to the returning venous blood that goes towards the body's core. Similar countercurrent exchange systems keep aquatic mammal flippers from losing body heat due to cold, deep waters, and peripheral heat exchange enables legs and feet to operate at near freezing temperatures for prolonged periods of time, like during a hunt. Thermal regulation. Small animals have limited thermal regulation due to their size and so must live in the subnivean environment, which is the layer between the fallen snow and the terrain. This is the environment where many animals that hibernate can be found since it provides insulation and protection from predators. Adaptive hypothermia in certain birds and mammals have evolved a unique mechanism of abandoning homeothermy for extended periods of time, and that allows their body temperature to fall. Some birds and mammals have an intense metabolism that requires daily food intake that nearly matches their own body weight. This is a difficult thing to support during cold nights, and most birds and mammals sleep at night to avoid predators. Torpor and hibernation. Daily torpor is an adaptive hypothermia mechanism that drops an animal's body temperature when asleep or inactive in order to prevent energy loss. Hummingbirds can drop their body temperature at night when food supplies are low. Body temperature and oxygen consumption, indicated by the red line, are higher when these birds are active during the day and can drop to 1/20th of these levels when food is scarce. Torpor lowers the demand on sometimes limited energy resources. Hibernation is the prolonged and controlled state of dormancy, common in medium-sized mammals in northern temperate climates. This behavior allows certain animals to meet the challenges of winter where there are food shortages in combination with low temperatures. True hibernators like ground squirrels or jumping mice and woodchucks are going to prepare for winter by storing body fat. Entry into hibernation is a gradual process where they undergo a series of test drops in body temperature. Eventually, the animal cools to near ambient temperature and its metabolism decreases to a fraction of its normal rate. Respirations may drop from 200 breaths per minute to 4 to 5 per minute, and the heart rate drops from 150 to 5 beats per minute. By contrast, animals like bears, badgers, raccoons, and opossum enter a prolonged summer sleep with little or no decrease in body temperature. This is not considered a true hibernation since these animals can awaken if disturbed. Bears can sleep for several months with their heart rate dropping from 40 to 10 beats a minute, but their body temperature remains the same.